This is the first YouTube of a series of 3 about calcium time series and how we can convert the time series that are recorded in, let's say, a slice with confocal microscope to extract from this time series time correlation that would lead to a graph representation and a better representation that will allow to extract statistics of a network, of the underlying network. So the plan of this ensemble of YouTube are the following. I'll spend some time now to describe what are astrocytes in the brain, their shape, interaction, the calcium bursting, the activity, and their diffusion, the connectivity that can be assessed by a diffusing dye. Then in the second YouTube, part 2 will be about calcium time series and the signal pre-processing that will allow to define connectivity and to reconstruct the underlying network from this correlated time series. And finally, I would like to insist on the notion of path coding, that is probably and possibly the neuronal code can be much more be represented by paths that consist of a succession of activated astrocytes rather than simply spiking events in a population. So let's start with the first part, and this will be now a biological description of known results. So this is to remind the classical pictures made more than 100 years ago by Ramon y Cajal, where you can see here astrocytes drawn really as a soma with this protrusion. And I think it has been for a long time not much appreciated how this protrusion could play a role and how they can be important, especially for the network. Okay, so the first, I think, important thing to note is that astrocytes occupy a single territory compared to neurons, where they can be all over the place. Here you can see by the color the astrocyte occupying one space and the neighboring one are located at the border. And this is how they pave. This is, I think, an accurate vision of how they pave the brain. So this is again as opposed to neuronal networks where neurons are really intermingling all the time, creating connections between them. This is opposed here with this reconstruction of astrocytes, where from calcium imaging you can see that they are really separated. And again, you can see here that with this anatomical view that they have this protrusion that can project, and not necessarily with directly to the most closest one, but they can project a little bit further away. And this is important because this could be the reason why they are forming heterogeneous, at least locally heterogeneous network and be connected maybe with specific cells. Maybe the genetics of this is unknown, but that's what we are going to be interested in here in these videos. So let me show you this. I think this is a nice reconstruction from Kyoshi et al., where you see here the neurons and then the astrocytes with these different colors and how neurons and astrocytes are intermingled. And also here you can see very well in 3D how the astrocytes territories are very well separated from one another. And the big tubes here was blood vessels. Okay, so with this in mind, we can now move on with calcium dynamic in the slides where you see here the bursting activity and astrocytes being activated here, but also the neighboring astrocytes also being activated spontaneously. Just to remind you, this is now very well known that it costs energy for this to function, but if you compare the amount of energy, at least for the resting potential, for astrocyte and glial cells here compared to neurons, they are quite cheap in terms of the energy consumptions compared to the action potential, 22%, synaptic transmission, 64, and so on. Yet, I think that we should really probably redo this calculation, account for example, of potassium regulation or calcium burst inside astrocytes. Okay, so they play a fundamental role for metabolic support, bringing the glucose and oxygen from the capillary to the neurons through complex pathway that I'm not going to go into detail. But I think they also play a fundamental role in potassium and regulation of potassium during neuronal activity. So what is also interesting is that depending on the demands, astrocytes are going to be more or less more involved if you have a larger demand compared to less involved if you have a less demand in metabolite, which is a proxy of neuronal activity. So how astrocytes are connected, this is again very classical biology that I'm bringing here for the next YouTubes. They are connected by gap junctions, which is a small molecule to go through passively, and notes that this gap junction can be at the soma, but most likely on protrusions that are connecting primary or secondary protrusion. And this is how the experience is done. You just inject a dye at a given astrocyte and you wait, and you see that the neighboring astrocytes are illuminated, confirming that the dye molecule is simply diffusing in the network, and this reveals how dense is the connected network. It doesn't say specifically here, although it can be seen that you have the impression that there are more fluorescence here at the center compared to the periphery, suggesting that there are more local connection here at this the small neighborhood of this injected dye. Okay, so we discuss here the astrocytes, how they are connected through these gap junctions. And what is interesting is that according to the region of the brain, here is the hippocampus, some neuronal, such as here the pyramidal neuron, can result in local depletion of the astrocytic density, suggesting that different region of the brain will be associated with a different concentration and organization of astrocytes. This is in opposition with this somatosensory cortex, where although there is an homogeneous astrocytic distribution, connexin 30 and 43 are highly expressed in the barrel cortex, but weakly expressed in the septal astrocyte. Again, suggesting, because the connexin allows the molecule, the passive molecules, to go through, and also the ions, that there are differential organization depending on the region of the brain. So we should maybe expect different types of functional connectivity between astrocytes. So in summary, for this first YouTube, we discuss here the astrocytic network that are responsible for metabolite collections and the delivery to neurons, and they are not only involved in this metabolic pathway, but also in uptake of neurotransmitter and extracellular potassium regulation, which can influence bursting and also epilepsy, of course. So this is again the challenge of the classical view that astrocytes were just simply providing nutrients and the environment for neuronal activity, to the concept that actually they can regulate the neuronal activity through multiple feedback. And the disruption, this is shown in here, the change, for example, in the gap junction, can be associated with a change in the behavior of the mice, and this is well summarized in this recent review by the Rouach lab. So there could be multiple astrocyte organization also, and this is understood also at the subcellular level, where there could be 3 types of distinguished region: the soma, the second, let's say, region, which is the proximal processor, and then the distal processors. And so the distal processors are involved in the in shaping the network of astrocytes. So in conclusion, there is a multi-scale organization, protrusion, primary processes, and soma. Astrocytes are non-overlapping territories, yet they could invade with very small protrusion or connect one astrocyte to the next one. They communicate through calcium signaling, although it remains unclear what defines these nanodomains. It involves IP3 receptors and calcium-induced calcium release from the ER, but really the understanding of how nanocompartments are really controlled and how calcium spread or not, it remains unclear. And so we are interested, and this is what we're going to see in the next YouTube, is to understand if there is a structure in this astrocyte that and form specific network. All right, I'll stop here, and I'll invite you to look at the second YouTube, where we are going to discuss the methods to reconstruct sub-network from calcium imaging in this in slices that shows spontaneous activity.